Hi everyone, in this video we're going to take what we've learned about transcription and really start applying it. So I want you to be able to identify coding versus template strands given a certain set of cues similar to what the cell would be doing, and we'll talk a little bit more about this. So you should be able to do this and feel pretty comfortable with this by the time we're done here. Given the gene, you should be able to determine the sequence of the mRNA that would be made. And you should also be able to determine the first few amino acids encoded by the gene. So this is where we're starting to practice using that genetic code that we've had before. All right? And then we're going to lead into mRNA processing. So there's three main aspects of mRNA processing that you have to understand and know. One is the 5' methylguanosine cap. The second is the poly A tail, and this is at the 3' end. And then we'll talk about differential, although your book calls it alternative, so you can, either of those terms, they're synonymous, differential splicing. And these are the main aspects of mRNA processing that's really important to go from a pre-mRNA after transcription to a mature mRNA that can be used for translation. And then after this asynchronous video activity, we will basically be focusing on translation over the next couple days on Wednesday and Friday. All right, so let's review what we've learned about transcription so far. We know that the regulatory regions, enhancers and silencers, they're what determine if the gene is on or off in any given cell. And usually these are going to be upstream. So if this is the gene start site, then downstream is down the gene from the start site. So this would be going in this direction, left to right here. And so usually, not always, but usually the enhancers and silencers are a little bit upstream, and this controls if that gene is on or off, right? And so my first question for you is, why is it important to control if a gene is on or off in a cell? Well, we know it's really important to control genes, gene expression, because that's what makes our cells different, right? All of our cells have the exact same genome, but a red blood cell is different from a muscle cell, which is different from a neuron, and so forth, because they are using different sets of those genes. They are turning on different genes. And some genes even need to be on in some cases, and off in other scenarios in the same cell, or on or off at different levels. So those enhancers and silencer regions and how they're controlled by transcription factors are really important to cell differentiation and cell activity. All right? But those are usually kind of further away, not too distant, but further away from the gene. And you'll talk a lot more about these in genetics, molecular biology, and developmental biology if you take developmental. Right. Then we also know that there's a promoter region, and this we have focused a little bit more on. The promoter region is basically the region that helps RNA polymerase know where the gene begins. All right, this is basically helping that RNA polymerase find the needle in the haystack, because remember, you have 3.2 billion nucleotides in your human genome, and only less than 2% of that actually codes for a gene. So the RNA polymerase not only has to find the genes in that 98% that are non-coding, but so they have to find that 2% of coding regions, but they also have to find the right start site, because if the transcription starts at the wrong place, your gene and therefore your protein is going to be messed up. So we've talked about the TATA box. So the TATA box and a few other GC-rich regions, these are right upstream of the transcription start site, and they bring the RNA polymerase to the correct spot. Right. And then when the RNA polymerase is there, we know that it transcribes the RNA coding region, and it has to stop at a terminator region. And we identified the terminator region basically usually with multiple A's or T's. So my next question for you is, why is it usually multiple A's or T's? All right, so think about that. You have some options in the Edpuzzle question. Think about that. Well, the reason is, remember, A and T, or A and U, if we're talking about mRNA, that binds relatively weakly because there's only 2 hydrogen bonds per pair of A and T, A and U. And so without many GC pairings in there, that is much weaker binding. And then usually there's something that has slowed down the RNA polymerase, whether it is the inverted repeat loop that basically acts kind of as an anchor to the RNA polymerase. But while it's paused, those stretches of A's or T's with only the 2 hydrogen bonds being weak, it causes the RNA polymerase to fall off, and that's what terminates or stops the transcription at the termination site. So the next thing is which direction. We also know directionality. So which direction does RNA polymerase read the template, and which direction does the mRNA grow in? All right, so remember, we're thinking about our DNA polarity. We have a 5' end and a 3' end. Which direction is the template read, and which direction does the mRNA grow? Well, we know that the RNA polymerase transcribes RNA. It reads the template from 3 to 5, right? The template's always read from 3 to 5, and the mRNA strand, or the new DNA, or RNA in this case, strand grows from 5 to 3. And so that's true here. That was true with DNA polymerase in DNA replication, and it's true with RNA polymerase making the mRNA strand. So based on that, we know that this bottom strand has to be the template because the promoter is over here and the termination site's over here. So we know that this gene is being read left to right, kind of in the direction I have it shown here. And since the template has to be read 3 to 5, the strand of DNA that is 3 to 5 from left to right is the bottom strand. Thus, the bottom strand has to be the template strand because it's being read in this direction, 3' to 5' towards the right. Right? And if that's the template strand, we know that the opposite strand is the coding strand. And so this basically is our set of cues to know which end the promoter is, which end the termination site is. Therefore, we know which direction the mRNA is made, in this case left to right, and the strand that is 3 to 5' in that same direction is the template strand. So this is important because, brief review from what we talked a little bit about on Friday, the different genes are transcribed in different directions. So we have chromosomes. We have 46 chromosomes, 23 pairs, and so there are different genes on each chromosome, but that means each chromosome has to have a lot of different genes on them. And they're not all in the exact same direction on that chromosome. So for example, one gene might be, gene A here is being made left to right on this chromosome, as we have it oriented here. Gene B is actually starts, the promoter would be over here on the right, and the termination on the left, so gene B would go right to left. And then GC back with the promoter on the left, termination on the right, so it would go left to right. Right. So with this in mind, for which genes is of these, gene A, B, or C, is the bottom strand the template strand? Well, we know that whatever direction it is being made in, the strand that is 3 to 5 in that direction is the template. So for gene A, 3 to 5 is left to right, which is the way the bottom strand is 3 to 5 in the direction that gene A is being transcribed, left to right. And so therefore, the bottom strand has to be the template strand. Same with gene C. It's being made left to right. The strand here that is 3 to 5 left to right is the bottom strand, therefore it's the template. For gene B, it's being made right to left, and the strand that is 3 to 5 from right to left is the top strand. So the top strand would be the template strand for gene B. So the answer to this one here, for where the bottom strand is, it would be genes A and C. The bottom strand is the template. All right, so the different strands can have different templates, all right, but this is where we can start to put together our cues, right? We know that the promoter, we can find the promoter region, and the terminator region is on the other side, so the gene has to be made from promoter to terminator. So in the case of the top one here, it's going left to right, promoter's on the left, terminator sequence is on the right, it's going left to right, therefore the bottom strand's the template, because that's the strand that is 3 to 5. Right? But we could have a different scenario, so let's look at the middle scenario here. Right, so in this case, the promoter is on the left, terminator is on the right, so we're still moving left to right, right? But now, the way the strands are shown, it's actually the top strand that is 3 to 5 from left to right. So the top strand in this case would be the template because we have to use our multiple cues. We know where the promoter is, we know where the terminator is, and then we know which direction the gene is being read and the RNA is being made, and the template is 3 to 5 in that same direction. And then we could have something like this, where suddenly the bottom here, the promoter's on the right, the terminator sequence is on the left, so we know this is actually being transcribed from right to left. And so which strand is 3 to 5 in the right to left direction? Well, in this case, the top strand. So the top strand, again, in this scenario is the template, the bottom is the coding. All right, so that's really important. So let's think about this and let's actually put it together with some real aspects. So here I have some DNA. I have the two strands. They are complementary, so when I have a T on one strand, I have an A on the other, and so forth, G and C. One strand's 5' to 3', the other 3' to 5'. So this is standard DNA. Now it's important to note that this is a very small fragment of what DNA would be. In fact, your chromosome, so that any DNA would be somewhere amongst hundreds of millions of nucleotide long chromosomes. So this DNA would be somewhere in the middle of that. And also genes are usually tens of thousands of nucleotides long minimum. So this is a very small kind of theoretical protein so that we can fit it on the page. But regardless, we have our two strands of DNA. And what I want to know, what I want you to figure out is which is the template strand. All right, so let's think about this together. Hopefully we're able to at least try and answer it, but let's think about it. So this is the exact same strand, but I've pointed some things out to you. We know what the promoter looks like. All right, so if we're back here, I know the one thing that I really understand about a promoter is there's the TATA box, and it's just a little bit upstream of the transcription start site. So I'm looking around trying to find the TATA box. And this right here looks like a TATA box. And even if you look at the complementary, it still looks like a TATA box, mix of A's and T's. So that is your promoter region. So I know this side has the promoter on the left. And then for the terminator, we're looking for that long sequence of A's or T's. And it's not a TATA box, so they're not intermixed usually. It's just kind of a long sequence. So over here would be our terminator sequence. Right? So with that in mind, I now know that this gene is being transcribed from left to right. This is really no different than when I just kind of labeled this as promoter and terminator, but now we're adding in the aspects that you already know about what a promoter and a terminator sequence look like. All right, so now I want you to recognize the promoter sequence with the TATA box and the terminator with A's, long stretches of A's or T's. All right, and so when we have that, now try to answer. So which is the template strand and which is the coding strand? Well, with this, we know that this gene, the way I have it shown here, is being transcribed from left to right, go from promoter to terminator. And so if it's going left to right, the template is the strand that is 3 to 5 in that direction. So the bottom strand would be my template because it's 3 to 5 from left to right. And the top strand would be my coding strand because it's 5 to 3 from left to right. So the bottom would be the template and the top would be the coding. And what I've given you is the start of the transcription site, just kind of the theoretical start. So this is where RNA polymerase would start transcribing this gene. TATA box gets it to the right place, and then the terminator tells it where the gene ends. So my next question for you is, with that in mind, knowing that this is the start of the transcription site, what would the sequence of the mRNA be? Well, there's two ways to think about this. We know that the mRNA is complementary to the template because the template is red, and then the mRNA nucleotides come and bind to the template strand based on base pairing complementarity, right? Because remember, the RNA polymerase is going to temporarily unzip the two DNA strands from each other, and that leaves the template strand available for nucleotide binding based on the complementary base pairing. Right, so when the RNA polymerase separates these two strands temporarily, a C is available on our template. That means a G is going to come bind. Then an A, then a T, which means an A will bind. A G, which means a C will bind. And then A, then G, then C, C. Since it's A, remember it's RNA, so instead of T, a U comes in. So U, A, U, G, G, and so forth. And we know that the mRNA has to be in the opposite orientation. So the template being 3 to 5 from left to right, the new strand would have to be 5 to 3. Right? So we can take a look at this here. And this strand would be our mRNA strand because this is complementary to the template and in the opposite direction. Another way you can think about this is we know that the mRNA is basically the same as the coding strand, with the exception that when the coding strand has a T, the mRNA would have a U instead. So we can also see that it's the same orientation as the coding strand. We know the top's the coding strand. So 5 prime, G, A, C, A, G, C, C, U, A, U, G, G. That also is what's going to be the mRNA. So if you're given a strand and you know the template strand or can figure out the template strand, you should be able to determine the mRNA sequence that will be made. That is one of the things that you will have to do on an exam. You'll have to find the template strand, you'll have to find the coding strand, figure it out based on the types of cues we've given you, and also be able to figure out what the mRNA sequence would be from either the coding or the template strand, whichever you prefer. Alright, so now next step, find the start site and determine the first 3 amino acids of this polypeptide. Alright, so remember, the start codon is the only codon you have to have memorized, and that is ATG if it's DNA, or AUG. Right, so I'm going to look down here, just like we did in our molecular modeling. I've found the transcription start site. Now I'm looking for my start codon. And remember, it's going to be in the coding strand, because the coding strand is the same as the mRNA. It's the mRNA that we use for our genetic code. So when you're using the genetic code, you're either using the mRNA or the coding strand, hence the name coding strand, to determine what the amino acids would be. So here's my start codon, ATG, so that's going to be methionine. And then GAC, that's my next codon. TGC, that's my next codon, and so forth. So hopefully you were able to use your genetic code. And what I want you to remember from this is to read the genetic code, you use the mRNA strand. All right, so you actually could have used the mRNA strand that we had here, except you would have to find the start codon and keep going with the mRNA strand. Or you can use the coding strand. And then you always find the first start codon following the transcription start site, and then you keep going until you get to a stop codon. So the first 3 here would be methionine, because methionine is the start codon, and then GAC codes for aspartic acid, and TGC codes for cysteine if you look at your genetic code. And then you could keep going. Actually, TAG, I believe, is a stop codon, so this would be a really, really, really short polypeptide sequence. But this is how your cell machinery identifies the start of a gene, identifies which strand is template, and then from the template it'll make the mRNA, and then the mRNA will be read and converted into protein by ribosomes in the cytosol. And that second part there is translation, which is the focus of Wednesday and Friday. So this just gave you a little bit of a taste of that. Okay. So once again, what you really need to be able to do is figure out coding versus template strand, given the cues that we've given you, recognize the data box, recognize the terminator sequence, and then be able to give a sequence of mRNA if you know the template and or coding strand of the DNA gene. So this is the transcription summary. I'm going to just show a quick video here, but basically you have the start site. RNA polymerase finds the transcription start site because the promoter region in the data box brings it there, and then it'll move and read the template from 3 to 5, making the mRNA based on complementarity until it hits the terminator sequence, and then the mRNA, the pre-mRNA, is complete, because we're going to talk about mRNA processing next.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box. An enhancer region may also be involved.
So remember, this enhancer region, that's where the enhancers or the silencers are involved to control gene expression, and that TATA box brings the RNA polymerase to the right place.
Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the TATA box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach.
We haven't talked much about transcription factors, but these are the proteins that are able to find that TATA box, and then they bind to it and then basically bring the RNA polymerase to that correct start site. So these are kind of the intermediates that identify our promoter region and bring RNA polymerase to the correct spot.
Complexes prepare the DNA for the successful binding of RNA polymerase.
This brings RNA polymerase to the correct transcription start site.
Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA template from the strand of DNA.
So you can see the RNA polymerase temporarily unzipping the two strands of DNA, right? And then the mRNA nucleotides are brought in by complementarity to the template strand. And then behind that, the DNA kind of reforms its double helix, and out emerges our mRNA strand.
Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates, and the newly formed strand of RNA is released.
All right, now we're going to talk about processing of that mRNA strand. So when that mRNA strand is first made, as RNA polymerase moves along the DNA and reads the template from 3 to 5, thus making the mRNA from 5 to 3, that initial mRNA that is made is a pre-mRNA. It is not ready for translation into proteins yet. It needs modifications, and there are 3 main modifications. And the first one is a 5' cap. Right? This is a methylated guanosine triphosphate cap. And here is the key to this. This guanosine, all right, just a standard G, like any other G in the nucleotides, but this is put on the 5' end, the very end. It's put on in the backwards direction. All right? So in other words, everything else here is 5' to 3'. The rest of this mRNA is 5' to 3', but this links 5' to that 5'. Right? So basically it links back to back. Right? So what do I mean by that here? In most of our nucleotides, remember we have our phosphate sugar linked to the next phosphate sugar, linked to the next phosphate sugar, and so forth. They're all in the same orientation, 5' to 3'. But this one actually is linked sugar phosphate. It's linked backwards, so the phosphate links directly to the phosphate of the other one. So this backwards guanosine at the 5' end really protects the mRNA from nuclease degradation. Our cells and bodies have a lot of nucleases. Let's think about that term right now. ACE means enzyme, usually an enzyme that breaks something down. So a nuclease would break down nucleotides or nucleic acids. And so if they're able to break down nucleic acids from the 5' end, it would just chop this mRNA, boom, chop the next one, cut the next one, cut the next one. But because it has this cap in the backwards orientation, our nucleases that normally degrade something 5 to 3 will not degrade this because it's protected with this backwards guanosine. Right? So what you need to understand is that this backwards 5' guanosine cap at the 5' end of our newly made mRNA, that protects the mRNA from nuclease degradation so that it has long enough to be read and made into protein during translation. It's also going to aid the mRNA in transport from the nucleus. All right, so this helps the mRNA get out of the nucleus to the ribosomes and the cytosol, where it can be translated into protein. And it also is going to help signal the ribosome to attach for translation. So there are basically three main purposes of the 5' cap. And this is what you need to understand for the 5' cap. That's essentially it. Right? Protection, transport from the nucleus, and ribosome attachment for translation. All right, then the second modification is the poly-A tail. Poly means many. A is that nucleotide, adenosine, and so the poly-A tail is added to the 3' end. Remember the 5' cap obviously was added to the 5' end. That's a methylguanosine cap. The 3' end gets these long sequences of adenines added, and this can be anywhere from 50 to 250. It's a pretty wide range. This basically has the same three purposes as the 5' cap, only it does it in a slightly different way. Right? So this is also going to help the ribosome attach in the cytosol. It's going to help facilitate export from the nucleus to the cytosol so the ribosomes can attach and translate the mRNA into protein. But it also protects from nuclease degradation. This it does in a little bit different manner. So remember how I said the 5' cap protects it from nucleases degrading it in this direction. Well, there are also nucleases that can degrade from the 3' end. And so the poly-A tail is not really preventing that. They can still be cut off one at a time. But let's think about this for a second. This region here, this is the important region that has the code for our protein. So if we add this long poly-A tail, kind of this long nonsense set of nucleotides of A's there, we've basically given ourselves a huge buffer. It'll take a long time, or quite a while, for nucleases working, cutting these off one at a time from the end until it gets to something important. And that actually is really good thing because, one, it gives this mRNA a long enough lifespan until that nuclease basically cuts into this here. Plenty long enough for a lot of protein to be made from this mRNA. But the fact that it's not protected forever and eventually the nucleases will start degrading this main part of the mRNA is actually a good thing because that means the gene that was turned on is only going to make protein for a relatively short period of time. Because the mRNA that was transcribed while that gene was on is relatively short-lived, it's only going to make protein for a small amount of time. And so the cell, if it keeps needing more of that protein, it has to keep transcribing that gene. But this also allows the cell, if it doesn't want that protein anymore, if it's changing its proteome because it's changing its activity, all it has to do is leave the gene off. The mRNA will degrade relatively quickly after it's made a few proteins, and then those proteins eventually will degrade as well, and now that gene is off and the cell has changed its activity. Alright, so back to the main take-home messages of this though. That poly-A tail protects the mRNA from nuclease degradation. It basically gives it a buffer of time, right? The cap protected it directly. The poly-A tail on the 3' end gives it a buffer where the nuclease has to chop through all these adenines that we added before it gets to anything important, and the mRNA is no longer able to translate and code for protein, right? But like the 5' methylguanosine cap, it also facilitates mRNA export from the nucleus and helps the ribosomes attach. All right, the final aspect of mRNA processing as we go from the pre-mRNA to the mature mRNA is splicing, all right? And so the genes, right? So here is our cartoon of a gene. We have DNA, and we have one strand 5' to 3, the other 3' to 5. One of these will be your template, one of these will be your coding strand, and so forth. But the gene has these short regions that don't code for protein, right? So they're called introns. The introns are non-coding sequences of DNA that are within the gene. So this gene might start over here and end over here on the right, and there are exons that code for protein, but there are also introns that don't code for the protein interspersed. All right, so when that DNA gets transcribed into mRNA, and then we have the 5' methylguanosine cap, we have the poly-A tail added, and remember this would be 50 to 250, pretty long there. These non-coding areas need to be dealt with. So my question for you is, which parts of these need to be spliced out and which needs to be connected? So are the introns going to be spliced out and the exons connected, or are the exons going to be spliced out and the introns connected? Well, remember, by definition, introns are the parts of the DNA gene that do not code for protein. So if the whole purpose of the mRNA is to create that code for one sequential protein, we don't want those intron regions in between. We only want the coding regions, which are the exons. So we have to splice out the introns and link the exons together. Right? And so here's our pre-mRNA, and basically these introns are going to be cut out and the exons are going to be hooked together. And so that takes our pre-mRNA to the spliced mature mRNA. We've gotten rid of the introns and we've linked the exons together. And so this actually happens with some machinery in the cell. We don't need to go into too much detail here, but basically what I'm trying to show is that the two ends of the exon, right? So this would be the 5' end of one exon and the start of the next one. So if we go back here, this would be basically this end hooking to this end. And this long sequence in the middle would be the intron. So all of this in light green would be intron. And some of these introns can be tens of thousands of nucleotides long, so they can be pretty huge. Right? So the end of one exon and the beginning of the next exon comes together, and that cuts out this big long looped part here. So we have this machinery, these proteins that do it, and while they cut out the intron, they've brought the two exons together and they just link them together. Right? And so that gets rid of our intron while holding the exon together. So basically the role of these proteins is to grab the two ends of the exons, bring them together, hold them together while the intron is cut out, and then just linking the exons back together with a covalent bond. Okay, so the introns are removed, exons are put together. Well, the next question you might have is why? Why go through all this? Why even transcribe all those introns just because, to cut them out? Right? And that's a really good question, but these do have purposes. This splicing allows for what's called differential, or as your book calls it, alternative splicing. Right? So if here we have our gene, and these blue squares kind of represent exons, so exon 1, 2, 3, and 4. We can link these together, where exon 1 is linked to exon 2, exon 2 to 3, and 3 to 4, and that is going to code for a protein that includes all those exons together. So that encodes a different protein than if, in this case, 1 still links to 2, but if 2 links directly to 4 and 3 is skipped, in other words, it's kind of removed with the introns, that's going to code for a completely different protein. So now we can go from one gene to encode multiple proteins based on how those splicings, how the splicing occurs, which exons are linked together. And this is important because we know we have about 23 to 25,000 genes in our human genome, but we make 75,000 to 100,000 different proteins because each gene can actually code for multiple proteins depending on splicing. So this is a different way of showing that here. So exon 1, exon 2 is the red, exon 3 is the green. If you splice all three exons together, you're going to have an mRNA that's bigger and codes for a protein with all three of these domains. But if exon 1 was spliced directly to exon 3, well then you would get this mature mRNA, which codes for a completely different protein that's lacking that kind of the red part, this whole huge part of the protein. And we know that if you change a single amino acid, you can change the fold of a protein and therefore its function. Well, obviously, if you're missing an entire region of that protein in this splice variant, this protein is going to fold and function very differently than this protein. Right? And this is just an example of this. This is a practical example. You don't need to know this directly. This is not going to be on the exam or anything. But I want to show the real importance of this. So Drosophila, fruit flies, basically have a gene called TRA. And TRA's pre-mRNA, well, it starts at the transcription start site and ends at the terminator, so all exons and introns are included every time in transcription. So that pre-mRNA has all the introns and exons. But this has two alternative splice sites. Right? So basically, exon A can bind to exon B, right? So include exon B, or exon A can bind directly to exon C. And if it binds directly to exon C, you get this protein here. Basically, exon B is missing. And so this creates a fully functional TRA protein. However, if exon B is included, there is what's called a premature stop codon. So when the mRNA is being read, we'll start at the start codon, and it gets to the stop codon in exon B. That means the whole rest of this is actually missing. So even though the mRNA is longer, the protein ends up shorter because exon B contains a stop codon. So that gives us a non-functional TRA protein. If you leave out exon B, that premature stop codon is not there, and the protein is made, the much larger version of the protein is made, including parts C and D. And this difference in TRA, whether it's the full TRA or the non-functional TRA because of the premature stop codon, is so important that it basically determines sex in Drosophila, right? If you have the full TRA protein, because of this splicing, this Drosophila will develop as a female. If you get the splice from A to B that includes this premature stop codon, that will become male. So obviously splicing is very important. Another quick example is a protein that induces apoptosis in our cells. Apoptosis is programmed cell death. That's a really important thing so your cells aren't immortal and cause cancer. There is a gene called BCL2. If it's spliced one way, the protein BCL2 causes apoptosis. If it's spliced a different way, it actually prevents apoptosis. So that's how important splicing can be to create proteins with different functions. All right, so in summary, we have transcription, and by now you should be able to kind of understand transcription, the process, and so forth. And then that gives us our pre-mRNA. And the first modification we have is adding the 5' methylguanosine cap for protection and removal from the nucleus, and so the ribosome can bind later on. Then we generate the poly A tail on the 3' end, also for protection and nucleus removal, export, I should say, and then the ribosome binding. And then we get the splicing that we just talked about. And once all of those things have happened, we now have a mature mRNA that is able to be translated. And that's what we'll talk about on Wednesday and Friday. And I just want to put this again into another context. So this is a theoretical gene that I basically made up, but this would be a great gene to have. So this gene is for the math polypeptide. So when this protein is made, this gene codes for a protein that basically helps you do math, right? But obviously you only want this gene on when it's necessary. So if you're not doing math, there's no reason for your cell to waste energy making the protein for the math polypeptide. So there are enhancer regions. Calculin is a transcription factor that binds when you are doing math. So this would be the enhancer that's turning on this gene when you're doing math, because you don't want the gene on if you're not doing math. But maybe you're really good at math and you don't really need that gene help, in which case you might as well save the cell's energy as well. So endor confidencia, right? So calculin, you're doing math, you need help. Endor confidencia, this is a transcription factor that gets activated when you're already confident. This would be the silencer. So if you're already confident, you're going to silence this gene because you don't need to make that math protein either. So the only time the math protein is being made is when you're doing math, you need the enhancer active. And when you're not already confident, you need the silencer not active. If the enhancer is active and the silencer is active, well, the gene's still off. If the silencer is inactive but the enhancer is also inactive, the gene is off. So the only time this will be on is when you have the enhancer active because you're doing math, and you have the silencer inactive because you're not already confident. All right, hopefully that makes a little bit of sense, but these are just planting seeds for what you're going to learn throughout the rest of your science career. So don't worry if it's not 100% concrete yet. But then we have our promoter region, which you can recognize with the Tata box. We have our terminator region, which you can recognize with the kind of long series of A's or T's. And that promoter region will bring us to a transcription start site. And then we have a translation. ATG is our start codon, and so these are the single letter of abbreviations for amino acids. But what I have in bold is the exons, and in lighter areas is the introns. And remember, introns don't code for protein, so they're not going to be decoded for amino acids. Only the exons are decoded for amino acids. So when this gets transcribed, we have to link our exons together, right? So here's where transcription starts. Link our exons together, get rid of these introns in between. So we end up with a mRNA that upon maturity will have a 5' cap. We'll have all of the exons directly together. So CCC-ACT is linked now to AUAA, CCC-ACT, AUAA, and so forth. All of these are linked directly together. Therefore, when it's being translated, it doesn't have to skip introns because they're no longer on this mRNA. So now, the mRNA can be used for translation. Here's our start codon. TAT would be the next codon, GAT, GAA, and so forth, until the ribosome gets to TGA, the stop codon. There we go. And you can see two last things. We have untranslated region of the mRNA. So we have the 5', this is the part ahead of the start codon that's not translated. UTR stands for untranslated region. Then we have the 3' end that also is untranslated because it's after the stop codon. That would be called the 3' UTR. And here's our poly A tail, 5' methylguanosine cap, exons spliced together. That is the end. This is our final mature mRNA. It's ready for translation. All right, thank you.